opening the door wide to new possibilities, hope of real breakthroughs and understanding more big ideas than we could have ever imagined. These are the biggest grants we've ever been able to. We are going to come out of this with some breakthrough ideas.
Focused solely on neuroendocrine tumors. You are not alone. We are all with you on the board.
So we have 2 more sessions. I'm going to introduce the next one, and then I'll have Dr. Woolen introduce his colleague. Thank you for those of you who are hanging in there. We have 2 more terrific speakers. Integrative oncology, for those of you not involved, Josh Mailman, who's now appearing on my screen, is very involved as well, and it's meant a lot to him personally, but also to the patients and the patient group he leads. So without any further ado, it looks like Josh Mailman is with us. If so, we have traveled from Germany at 3 o'clock in the afternoon, at whatever that was, in this morning, and now it's 6 a.m. in Australia?
7 a.m. actually.
7, okay, I exaggerated a little bit. So thank you for getting up with us, Josh, and welcome. I guess it's not still World Net Cancer Day there, but it still is here, and we are delighted to have you with us. We did a Q&A earlier. We had more questions. RT and Theranostics in particular, and we said, Josh will be with us. He'll give us the latest from the World Congress, which just concluded in Australia. So without any further ado, let me introduce and turn it over to you, Josh, to give us a quick update, and then if there are any questions, I'll relay them to you through the mic, okay?
All right, sounds great. Can everyone hear me okay? All right, so welcome to Melbourne. I've been here for a week, and part of this has been for the World Congress of Theranostics, and this is the 4th one. It just concluded on Wednesday here, which would be your Tuesday. And we had, I wanted to take you through, it really was a 3-day conference and actually a day pre-Congress with patients and a celebration of 20 years of PRT at the Peter Max Center. I was lucky, Professor Hoffman, Michael Hoffman, is one of the people who invited me here to Melbourne, and they did a preclinical spotlight of looking at what was interesting at the conference. And I will shorten it a little bit because it was 72 slides of interest, so I'll try not to go that far, but I just want to let you know, we look at this as we fight alone, but actually we fight with many people around the world. This is a quick chart of where the over 370 attendees for the Theranostics World Congress came from. You can see the predominantly from Australia, but a good contingent from the US, a large contingent from Germany and India, places where PRT is very commonplace, but from around the world, South Africans from Brazil, we have people from Poland, New Zealand, obviously, since they're close by. Gives you an idea of how international this is. This was a record for the number of abstracts that were submitted for the Congress. We had 124 abstracts from 26 countries. The abstracts and actually the videos will be available online, but to give you an idea of what the abstracts look like, I made a quick...
Oh. Okay.
Are you still there?
We're still here. You're back.
Okay. Sorry to have lost you. Where did we lose me, by the way?
You were just about to shift from the pie chart slide to giving us an example of the abstracts.
Oh, okay. So actually, there's a link of the abstracts that you can actually see. So you can actually pull them down, all 124, at least the titles. They'll be available online soon. And just a brief reminder, Theranostics is a way to have a diagnostic pair with a therapeutic pair. And the first World Congress was really devoted to gallium-68 and PRRT. And really, it was all pretty much devoted to NET and how to use a gallium-68 generator. The gallium-68 generator was really developed a little over 10 years ago, and at that time, there were very few in the world, maybe 5 or 6, and one day was devoted to teaching nuclear pharmacists how to do chemistry. It was orchestrated by Richard Baum, who couldn't be with me today, and Frank Reusch, also of Germany at the Mainz University, and included a practical day on actually how to use a generator. The Second World Congress started moving a little bit farther. We started talking much more about dosimetry. We started to look at new targets where the First World Congress really was all on nets. We started to see different things being used with gallium-68 and some of the other novel radiopharmaceuticals. Started to see the first time of using things like gallium-68 for pulmonary disease. It was actually co-run by Baljinder Singh here and of PGMER in India, and Michael Schultz from the University of Iowa, who works with Dr. Auditorium there. The third one was many of you, some of you in the audience I know came to the third Theranostics World Congress in Baltimore last year that was sponsored by Richard Wall at that time of Johns Hopkins. And that was really an inflection point where we started to see gallium-68 and being used with other targets. A third of the conference was on prostate cancer. So to give you an idea of what, how this Congress shook out, where the first one was all nets and nothing else, and the second and third started to introduce other diseases. What we had here was one day devoted to nets, one day devoted to prostate cancer, and one day devoted to novel theranostics or new ideas in the use of theranostics. I'm going to give you the major observation because, as I said, there were 124 abstracts and 20 plus hours of talk. So trying to get that all in the next 9 minutes will be next to impossible, but I'll do my best.
Just so you know, Josh, we're not seeing the slides too. We're just seeing you.
Oh, you're not seeing the slides. Great. Let me re-share the screen right there to make sure that we can. Sorry that you're not seeing the slides, but you will in a second.
But do you feel like you're in the movies? Because you do look rather large.
Is that better? Can you see it now?
Yeah, we got it.
Great. All right. Let's see if so here are my major observations.
Hey, Josh. Josh.
Yeah.
We're trying to be a little better about defining terms. Could you define theranostics for us, please?
Sure. Theranostics is the idea. It's therapy plus diagnostics. So if you look at it, that's theranostics. It's using, if you can see it, you can treat it. So the idea is that you have a diagnostic nuclei followed a diagnostic peptide that can target a tumor, and then it actually will link to a radionuclide that can be actually imaged. And if you can image it and see the tumor that way, you replace the diagnostic radionuclide with a therapeutic radionuclide that would have more energy to try to kill and disrupt the cell. So that's the theory behind theranostics. And neuroendocrine tumors lend themselves really great to this because they have such heavy receptor binding that it's very easy to get a target that you can bind to using, for instance, octreotide or various different versions of octreotide, whether dotatate or dotatalk or dotanoc. What we also see is that they're now not with peptides, but with things called antigens. You can really target prostate, for instance. So prostate-specific membrane antigen is something now is a new theranostic pair. And I think that's what we looked here, we really saw. We saw more places for theranostic. Let me just see if I can load that up. Well, we saw really new pairs. Gallium-68, we know as dotatate just got released the NetSpot, which is the gallium-64 nets that is FDA approved, but it's more than that. There's A lot of new targets that are coming, JR11, which is being worked on Memorial Sloan Kettering. There's some other pairs that don't use the somatostatin binding. This is copper 64 and 67, and I'll show you some images of that as well. I know one of the things that worries a lot of patients is that with these new diagnostics that people might not be able to understand or read them well. And one of the posters here was by Bonnie Clark from the Society of Nuclear Medicine and Molecular Imaging on national reader training being made for gallium-68 labeled imaging with dotatate and dotatalk and PSMA. I won't have dosimetry is the idea that we can figure out, or we try to figure out, the maximum doses that are going not only to the tumor, to be healthy cells like the kidney or the bone marrow. And while many places practice dosimetry, I think 2/3 of the centers here practice dosimetry routinely for NETs patients. It's hard for standard patient care because of its time-consuming and costly. And so there was a lot of debate here on if we need it, how we need it, and when we need it.
Dosimetry is? Yes. No. Okay, Josh, dosimetry, please, the definition.
Dosimetry is the idea of dose measurement. But the idea that when we put in a radionucleide into someone's body, we can actually trace where it goes and calculate how much is getting, how much actual activity is getting to a tumor. We talk about, giving someone 5 gigabecquerels or 20 millicuries, but that's the dose. What happens is, as it circulates through your body, you end up excreting or peeing out a good percentage of it. So the question is, how much went to the tumor, and more importantly, how much went to rate-limiting organs like the kidney? And if we can measure that, we can increase the dose or decrease the dose to make sure that the organs of interest, like the kidney and the bone marrow, don't get over-treated, because that is in fact what may cause other complications down the road. So the idea of dosimetry is always done during clinical trials before we come up with what the maximum dose is. But the question is, in working towards personalized medicine, do we need it with every time we do a dose, or can we get what is a standard average and do 4 doses of the same type and know that pretty much everyone's protected and getting a decent therapy dose? And I could actually spend an entire day talking about dosimetry, and half of a day here was, so I'll try not to. The challenge is dosimetry is very costly to do. It's a big physics question. It requires a lot of scans, pre-planning and post-planning, to see if your pre-planning worked. And it remains something that, without getting a randomized control trial to understand if we dose closer to dosimetry max, if we get better therapeutic outcomes. So it's a topic that will continue to be discussed over the years and over the upcoming years. And one other point that I made in some of my presentations here, the one that I gave, is, it's been 20 years since we had the first PRRT with Y90 in Europe, and 20 years since the first PRRT in Australia. But we still don't have an approval anywhere in the world, and I'll talk to that in a second. And this is largely because the nuclear medicine, lack of a better word, industry, or at least the academic centers, didn't really do randomized control trials, so it was very hard to get a registration. And that was my big point. Actually, we've treated thousands of patients. There's guidelines written, international guidelines on how to do PRRT. It's selectively available based on geography. And as of today, today, November 11th or 10th, we have no approvals anywhere in the world. And that may seem surprising to a lot of you, because I actually did a Facebook poll, and 76% of the people who responded felt that PRT was approved somewhere else in the world. Actually, if you take a look at it, it was actually nearly 90% felt it was approved somewhere in the world. And actually, the first approval is likely to come in the United States by the end of this year. So it's available as compassionate care and under institutional studies, but it isn't available as a commonplace. And as easily as the institutional studies and compassionate care have happened, they can go away with lack of funding and or what happened in the UK last year, where the actual funding for compassionate care, because it wasn't an approved therapy, was taken away. So going towards approval is a very big thing for this community. The big news, and I know that you've already, I believe Dr. Wolin is there and probably already talked about this. We do have a phase 3 trial that was in midgut. This is the NETRF trial that's been talked about a lot. There was new data presented, and I know this is an extremely busy slide, but I'm sure Dr. Wolin already talked about it, and how much better those that were on the treatment arm were doing over what the control arm was, which was a double dose of octreotide LAR. So I won't go into this because I'm sure you saw that. And this was also the big new announcements here were the quality of life initiatives, where the quality of life looks so much better on the treatment arm versus the non-treated arm. So that was a real highlight here, because this is a randomized control trial and this is what is used for registration. I'm going to go through, so a lot of what happened in getting PRT to market or doing was that places here, like in Australia, this actually slide is from Perth, they got the idea of, in the lab, of combining PRT with capecitabine and temozolomide, which is a chemotherapy agent. And they got a bunch of complete responses here. And they were, and everyone was doing really well under this particular treatment regime in Australia. And they also noted that their toxicity, because they treat people a lot earlier here, they had 0% of acute leukemia on all the patients they were treating, which is pretty much reported close to what some of the series that we've had in the US have been, and what NETR1 has reported so far, although it's pretty early in the NETR1 data set, and different than others. But many people found it hard to believe that they could get that good of a result. And so what they finally did was do what is a really great thing, is actually have a randomized control trial that is having 2 arms that compares a CAPE arm or CAPE-TEM arm to a with and without PRT, to see how that impacts both things like complete response and also toxicity. So that trial is ongoing here and enrolled, which is great. And this is where it took about 17 years for the first randomized control trial of LU-177 and dotatate. They did much quicker in getting this from something that had promising results in the lab or in trial for individuals to actually randomized control trials, so they could the word. I did talk about other, and I'm not going to go into great detail, but to give you an idea, this is what a copper 64 image looks like. This is actually something that doesn't rely as much or at all on having a somatostatin binder. So this might be very good for people who are who don't have type 2, and this is a receptor type 2, and this is in the early phases right now. And this gives it in comparison to what happens with comparison to gatate is what they call dotatate here. And what happens is gallium-68 really washes out after an hour or two, And the copper-64 can stay and really target the tumor, so you get a really great idea of exactly where the tumor is, and you don't get all this other noise of things that really aren't tumor. So you can really see it much better, and it turns out it has a Theranostics pair as well. What we see for, if you think of gallium-68, this is actually neuroblastoma. So a lot of what we learn with PRT and finding other receptor targets is that there are other things that we don't normally think of as in the NET world that really are. So this is a child that was able to be scanned with gallium-68 DOTA-NOC and then treated as well. We saw some, I don't have their slides, but some amazing results of what those look pre and post treatment. There were disruptive technologies talked here. We talked a little bit about, this is actually prostate cancer. This is gallium-68 being used in prostate cancer. Mostly you can see a lot of the bony metastases that you really can't see otherwise with prostate cancer. And there were at least 2 agents, and actually the image of the year for the best radiology image was actually done by Stanford, and this is using various different agents that are available for imaging prostate cancer. And you can see here the 2 that really can show up some new things are gallium-68, PSMA-11, and RM2, which are the 2 newer targets that were talked about here. So, and this gives you an idea. This is also a German slide of here's someone who has complete remission from prostate cancer using a Theranostics pair with gallium-68, PSMA-11, and the pair of lutate. So what started pretty much predominantly as a therapy for NETs is now used for other things. This is from Peter Mack. This is a patient who had tons of bony mets, and pretty much after a treatment, 3 months, you can see what happened. It's very exciting. So
Josh, we're going to wrap it up in just a minute.
This is actually, even now we can see PSMA in breast cancer and also HER2. So what started as something for NETs, and this is a paper from South Africa, has now advanced into something for other diseases as well. Let me see if I can, again, this is HER2 for people who are HER2 positive breast cancer, that this is being done in Vienna. And the last thing that I want to touch on is, you know, it's been 20 years and we don't have any actual approvals, and why is that? And we actually had a regulator come talk, and they trade off, you know, quality, safety, efficacy versus and not really finance. And they need clinical studies to really understand that the benefits outweigh the risk, and that the uncertainty around risk and benefits must be really, really small. And that you need a randomized controlled trial to understand the counterfactuals. Would these people have done as well on either other treatments or on no treatments? And this regulator from Europe brought up that they've had theranostic pairs, or some not in NETs, but that looked really good for 20 patients. And then when it was expanded to 200, it turned out it wasn't effective. But they also understand that with rare diseases and things where it's rapidly going, that there's going to need to be new pathways to figure out how to, and the cost actually of doing a clinical trial, and that they're going to have to actually rethink how the regulators work with randomized control trials to keep them both cost effective and timely. I think. So again, we treat many, but we don't, because we don't have randomized control trials done and registrations done, we don't treat a lot of people, and we need to get that done. One last point is clinical trials aren't cheap. Just to enroll in a clinical trial and track you is $50,000, regardless of what the cost of the drug is or how long you need to be followed. And that adds up when you're looking at doing 200 patients. As a group, we're going to need to move faster and smarter. And one of the points I was trying to make is we need not to repeat the past of having 20 years of a drug being used but not really getting registered. And with that, the Theranostics World Congress website, as you can see, is up there. That is a look at the new Peter Mac campus that just opened for cancer research and cancer care. It's a $1 billion facility. I'm hanging out with. I know I've talked maybe a little longer, but I hope that was interesting and informative and that you guys found it interesting.
Terrific. Let's hear a round of applause for Josh.